Hi everyone, it's Dr. LeBlanc with a linkage mapping problem. Tri-hybrid crosses are a common strategy used to create genetic maps. In this example, we're going to use three Drosophila genes on the same chromosome for body color, eye color, and wing shape. The two alleles for body color are going to be black, which is the mutant, and wild type, which is gray. Purple eyes, which are the mutant, and red eye color is the wild type, and wing shape is going to be vestigial wings for the mutant and normal wings for our flies. Remember that if the fly gene is designated with a plus sign, that means it's the wild type allele. So a cross between true breeding strains that differ with regard to three alleles is the first cross that we set up. Here's our true breeding, black-bodied, purple-eyed, vestigial-winged female mated to a wild-type male. The F1 that got produced are going to look like this. They're going to look wild-type, but we know that their genotype is actually heterozygous at each genetic locus, with one wild-type allele and one mutant allele. We then set up a test cross between one of these F1 females to a test cross male, and the test cross male is always homozygous recessive for all the alleles. So for this gene, it has the two black alleles, vestigial for this gene, and the two purple eyes for this one. Okay, so we're going to collect our data from the test cross. We know that if the genes had assorted independently, all 8 combinations would occur in equal proportions, but we know that these genes are linked together. And so the combinations of alleles that show up in the test cross are going to be skewed based on the distances between the genes and what the two parental combinations of alleles were. So the parental phenotypes occur the most frequently, either the wild type or the mutant, all 3 mutants. And that's true. Here we have 402 for the 3 mutant alleles and 421 for all 3 wild type alleles. Those are the most common. The double crossover phenotypes occur least frequently, and that's indicated by the red arrows here. Only one of this combination and 2 of this combination of alleles. And the single crossover phenotypes occur with intermediate frequency, and that's indicated by the green arrows. Those are all the rest of the flies. Question number 1. What is the order of the genes? We need to look for the odd man out in the double crossovers. So remember, our unrecombined parental combinations of alleles are either all wild type or all recessive. The lowest number of group combinations are the 2 double crossovers here, only 1 of this fly and 2 of this fly. So here we have 2 recessive alleles for body color and vestigial wings, but a wild type allele for eye color. So here's the odd man out. Here, it's the purple eyes. Let's see if the other double crossover gives the same results. Here we have 2 wild type alleles and the mutant allele for eye color. So yes, the purple eye color is the odd man out. So the gene for eye color then is placed in between the body color and wing shape. So do we have that answer here? We have one with purple eyes right in the middle. So we have a black body, purple eyes, and vestigial wings. So hopefully the answer is A. Okay, so that's how to determine the order of genes. How can we calculate the distance between the genes on the ends? Well, we need to look for recombination events between the 2 genes on the end. So we would need to find a wild type allele on one end for body color and a recessive allele for vestigial wings on the other end of this large genetic locus. And that's what we see in this combination. Okay, we have plus here and minus here. This one does not look like it's a recombinant between these two because they both have wild type allele, but we're going to come back to that. Here we have another wild type allele and another recessive allele. Here we have the reciprocal recessive allele and a wild type allele. And again, we'll come back to this, and another recessive and a wild type. So we're going to add up 63 plus 27 plus 31 plus 58, and then divide by the total number of flies. So we have this possibility here and this possibility, but we're not quite finished yet because we need to consider the double crossovers. So although this combination of alleles is the same as one of the parental types, and this combination of alleles is the same as this parental combination of alleles, we actually know that there were 2 recombination events in order to come up with this combination of alleles, with the purple mutant allele in between 2 wild type alleles. So we actually had to have 1 recombination event to get to be body color, wild type body color, and purple eye color, and then another recombination event to get back to the vestigial, the wild type wing shape. So we actually had 1 recombination event and then another recombination event. So we need to add up 1 plus 2 and then multiply these by 2 to get the actual distance between the 2 genes on the end. So it's got to be this one. We have 63 plus 58 plus 27 plus 31 plus 2 times 1 plus 2 times 100 divided by 1005 equals 18.4 distance. So that's the map units. That's the distance between the black body color and vestigial wing genes on either end of this locus. Okay, so what about the distances in the middle? What are the map distances between the middle gene and each of the 2 on the ends? Well, we need to figure out when there's a recombination event between the black gene and the purple gene, the body color and the eye color. Well, this doesn't look like a recombination event because they're the same combination of alleles that we see on one of the parental chromosomes. This one does, though. Here we have wild type and here's mutant. And this one does too, wild type and mutant. And this one does too, mutant and wild type. And so does this one, mutant and wild type. But not this one because they're both the mutant alleles. So we need to add up 1 plus 27 plus 31 plus 2 to get the distance between the black body color gene on one end and the purple eye color gene in the middle. So here we have 1 plus 27 plus 31 plus 2 equals 100. So the distance between B and P is 6.1 map units. For the distance between P and V on the other end, it's the rest of the combinations. Here we have vestigial mutant and wild type for purple eyes. So we're going to count this one and this one, not this one, not this one, this one, and this one. Anytime we have a combination of vestigial and purple alleles that's different from what the 2 parental combinations were. And so if we add those all up, multiply by 100, divide by 1000.5, we get 12.3 map units. So our answer is B, 6.1 and 12.3. So which is the correct map for this? Well, this is it. We came up with 18.4 map units between the 2 genes on the ends. We have purple in the middle. 6.1 map units between the black body color and the purple eyes, and 13.3 map units between purple eyes and vestigial wings.